All right, let's go ahead and get started. Um, One quick announcement is the snow that's coming in. It seems like Richmond's going to get a little bit more snow than Charlottesville. Again, for if you don't remember, I'm commuting in from Richmond every day. Um, there's a likelihood that Richmond's going to get a fair amount of snow. It seems like predictions are changing from day to day. But in case Richmond gets closer to what some predictions are, like a foot of snow, what I'll probably end up having to do is I'll probably end up having to stay in Richmond on Thursday. And if that happens, what I'll do is I'll post Thursday's lecture, a previous recording of Thursday's lecture online, and I will make the quiz. We'll still have the quiz. I'll make the quiz accessible for the entire day, but with a 10-minute limit. And so you can take it at your leisure. But again, honor code still applies. And so please make sure that please make sure that you do the quiz for one, that it'll be open throughout the day. I'll have it open until midnight. But this is just in case that the snow is significant on Thursday in Richmond and I can't make it in. Do you guys have any questions about that at all? Yeah. Yes. Yeah. As well as looking at other people's computers. That too. Um, yeah. Any other questions at all? All right. We can go ahead and get started then. What I'm going to do is I'm taking a step back a couple slides for us to recap where we left off. So we spent a lot of last class talking about gel electrophoresis. I'll take an even bigger step back. Big picture of what we're trying to do here. We are trying to, having discussed proteins and amino acids in the first unit, having discussed a variety of families of proteins, we're now taking a moment to try and explore how proteins are measured, how they're analyzed in a research or clinical setting, and leveraging the protein biochemistry to do so. So we talked about gel electrophoresis, where we're able to separate proteins either by their mass or by their charge, or in the case of a 2D gel, separate them by their charge and then separate them by their mass. And so, excuse me, in the gel, oftentimes a polyacrylamide gel, these proteins can be stained to look at total protein content. We talked about a couple of stains, Kamasi blue and Cipro ruby. But the question remains is, if you're not interested in looking at just total, all the proteins in a gel, what if there's one protein, one specific protein you want to look at? And so that's how that was our launch pad into Western blotting or immunoblotting. And so whenever you hear immunostaining, immunofluorescence, immunocytochemistry, whenever you hear immuno, think antibody. Antibodies are what allow you to detect one specific protein. And so because antibodies cannot access these proteins that are embedded in this polyacrylamide gel matrix, we have to try and make them accessible in a certain way. So Western blotting, what we do is we run our SDS page gel that is depicted in the image to the top here in the right column over there. And you typically can't really see all of these bands here. You can typically see in the very first well, there's usually what we call a ladder. It's basically this like mixture of proteins that have very specific molecular weights so that you know what the molecular weight of your protein is when you're running a gel. Um, but we'll run our samples down a gel via SDS page. And because these proteins are not accessible to an antibody, what we end up having to do is what we call transferring. We'll transfer them onto a membrane. Either it'll be made from nitrocellulose or PVDF, and it'll be on the surface of that membrane and accessible to antibodies. And so what we'll end up doing is depicted in the second image below here. Oh, forgive me. Depicted in this image right here, we'll have this almost sandwich where we have sponges on the outside, filter paper, and then our gel and the membrane in direct physical contact with each other. That physical contact is important because you're running you're running a current here across this gel and this membrane. And what's really important is that current always flows to the positive end. Current is the flow of electrons. Electrons are negatively charged. Negatively charged electrons are going to be attracted towards the positive end. And so they're going to move towards the positive end. And I made a joke or a comment last time where if you switch them around and if you have your, I'm going to move over here. If you have your negative end on the membrane side, when I say membrane side, it's just between the gel and the membrane. If you have your negative end on the membrane side and the positive end on the gel side, you're just going to run your protein off that gel and your sample will disappear. Speaking again from personal experience. So you want to when you're looking at relative to just the gel and the membrane, you want the membrane on the positive side of this sandwich, and you want the gel on the negative side so that it's running, the current is running towards the positive end, and you're running your proteins onto your membrane. And so once that happens, now you can stain using antibodies. So you run your gel, you'll transfer onto a membrane. They're on your membrane now. And what's important is that we're going to block using some blocking buffer. It's usually some abundant protein that occupies non-specific sites. That is going to be really important because, again, like I said last class, proteins are naturally sticky. And so you want to occupy these non-specific sites that do not inhibit specific binding of antibodies. And so once you block, you'll then wash and add a primary antibody, an antibody that's against your protein of interest. It's going to be, we usually, when we talk about protein antibodies being against some protein, the language that we use is that it is, for example, if it's an antibody against actin or tubulin, we would say it's anti-tubulin or anti-actin. And the species of the antibody is really important because the species will determine what secondary antibody you use. The secondary antibody is going to be anti whatever the species of the primary was. So if your primary is rabbit anti-actin, your secondary is going to be some other species, goat anti-rabbit. It's going to be anti the species of your primary antibody because it's going to be just an antibody that's generic for antibodies from that species. So I can try and write this on the board. Primary, shorthand for that is like 1 degree. So primary is rabbit anti, I'll just say tubulin. Your secondary would be of commonly used species is goat. So I'd say goat anti-rabbit antibody. Typically, the antibody class that's used is called IgG. So we would say it's goat anti-rabbit IgG. I'll write this over here as well. And this secondary antibody is usually conjugated with either some fluorophore or some enzyme that reacts with a substrate to produce a color change. And that'll allow us to be able to image and see the bands on our Western blot. And so what this looks like is, excuse me, on the left half here, we have Kamasi blue just staining again. Kamasi blue just stands for total protein. If you have a cell that you've exposed to some sort of lysis buffer that releases all of the proteins in the cell, you would have every protein stained in that gel. The second lane here, we have a purified protein that was purified via chromatography, both in lane 2 and lane 4. And again, lanes 1 and 3 are just total protein in some lysed tissue or cell sample. But Western blotting allows you to use antibodies to detect for one specific protein. So on the right half here, there's a Western blot where they were able to simultaneously stain for 2 markers using 2 antibodies, one for ATPase and one for thyroidoxin. When we talk about antibodies, we usually associate them with our immune system because there are immune cells that are uniquely tasked with producing antibodies. Those cells are called B cells. B cells are the antibody producing cell of our body. And so we can think of antibodies as being as almost a receptor for some sort of typically in the context of our immune system as some sort of foreign material or foreign molecule. The way we refer to those foreign molecules or those molecules that antibodies bind to, we refer to them as antigens. So for our primary antibody, in this example that I've written on the board, the antigen that this antibody binds to is tubulin. And so our primary antibody binds to the very specific antigen of interest. On the previous slide, there were the Western blot that we looked at here. The two antigens that these antibodies recognize are ATPase and thyrodoxin. The secondary antibody recognizes and binds to the species of our primary. Again, secondary here is goat anti-rabbit IgG. So it binds to that antibody. That's an IgG antibody. We have 5 flavors of antibodies. We'll talk about that next week. The one we'll focus on for today is just called IgG. And so secondary binds to the primary antibody based on what species it is. So again, for example, here we have another example where the antigen of interest is PKA, protein kinase A, and it's specifically its catalytic subunit. We talked about in the previous lecture series that cyclic AMP promotes the regulatory subunit to dissociate, and we just have the catalytic subunit. So this antibody, the antigen of interest, is PKA's catalytic subunit. The secondary antibody, because that's our primary is a rabbit, our secondary is goat anti-rabbit IgG, goat anti-rabbit antibody. And it's conjugated with an enzyme, which means there's a substrate that will expose it to that produces a color change that you can then image and see your bands on your membrane. Antibodies can come in different flavors. What you may have seen in either clinical commercials or seen in some of the journal articles you may have been reading as monoclonal antibody therapies. When you hear monoclonal antibody, what that refers to is how many parts of the antigen the antibody is binding to. So for example, if there's an antibody, for example, if my hand was an antigen, okay, we refer to the whole molecule as the antigen. We'll talk in a couple slides about how antibodies are made, but if an antibody was being created against my hand, if this was the pathogen, there would be different parts of my hand that would be recognized by different antibodies. There might be one antibody that binds to the last metacarpal on my thumb. It could be on the second knuckle on my pinky. These are called epitopes. These are specific sites at which that are recognized by antibodies. And so monoclonal antibodies is a cocktail of antibodies that bind to one and only one epitope. So monoclonal antibodies could be an antibody solution that binds to specifically the epitope of the nail on my pinky finger. Those antibodies won't bind elsewhere on my hand. They recognize this specific epitope. So the whole hand is the antigen. The specific binding site that's recognized by these antibodies are called epitopes. Polyclonal antibodies is a mixture of antibodies that bind to multiple epitopes. So there might be within a given tube of a polyclonal antibody mixture, there could be antibodies that recognize the nail on my pinky, the last knuckle on my thumb, just different parts of the antigen that are all recognized, but different epitopes. And so we have, the reason why it's called clonal is because there's different clones of B cells that secrete these antibodies for each epitope. I'll stop for a couple of questions here. I'm going to go back there and then up here. Go ahead. Yeah, great question. Okay, so last slide. We have ATPase and thyrodoxin that were stained simultaneously. If your ATPase antibody is rabbit anti-ATPase, if your thyrodoxin is rabbit antithyrodoxin, you're going to run into issues here for the very question you just asked, because you're not going to be able to distinguish what signal is what when you're imaging here. So usually what we'll do is we'll have different species for these antibodies, where perhaps this is a rabbit antibody, rabbit anti-ATPase, and then this is mouse anti-thyrodoxin. And then you can have different secondary antibodies. In the case, if we use fluorescent markers that have different wavelengths, and you're imaging on different channels for those different wavelengths, you can have, for example, down the hall here, we have an infrared reader for Western blotting. There's two wavelengths we use. We use 700 nanometers and 800 nanometers. You're measuring these two things. You can have one wavelength for your goat anti-rabbit, because this is rabbit anti-ATPase, and goat anti-mouse at a different wavelength, and you can measure them separately, but staining simultaneously. And so if you're staining for multiple things at once, you cannot have the same species for these two antibodies. You would be surprised how many times grad students need to be reminded of that. And so it's really important to make sure that these antibodies are different species if you're staining for two things at once, like in this Western blot. Yeah.
So seems like polyclonal would just be better in this scenario if you had polyclonal, right? So
That's a great question. So it depends on what scientific question you're answering. So if all you care about is just overall expression of some protein, polyclonal is going to answer that question. It's also cheaper. You also have to spend less money. Monoclonal antibodies can oftentimes be on the order of $500 to $600. Polyclonals can be on the order of around $100. And so it's usually cheaper, and it'll help you answer the question. So polyclonal would be fine. If there is a specific part of that protein that you're interested in to see, is it expressed or present, or in the case of kinase activity, like with protein kinase A, you can have antibodies that measure phosphorylation of specific parts of that protein. You could answer the question with a monoclonal, only with a monoclonal antibody, is this specific site phosphorylated or not? And so it depends, using monoclonal versus polyclonal depends on what question you're trying to answer. Yes.
so with the second antibody also bind to antibody, like you kind of antibody for same.
Sorry, say that one more time.
The second antibody, secondary antibody, also bind to other antibodies of the same species.
Could the secondary antibody bind to another antibody of the same species?
Antibodies of the same species that's trying to stain, but then
Different, like, are you saying different isotype as in, like, instead of IgG, it'd be like a different kind of antibody? Yeah, so it would not bind. So great question. When these secondary antibodies are raised, I shouldn't say raised, that's going to be confusing. When these secondary antibodies are developed, they're specific to IgG. There are the reason why I was trying to get a little bit deeper in your question is because there's different flavors of IgG, but to the question you're just asking about was like different different antibody families of IgM instead of IgG. So your secondary antibody is specific to IgG, and I'll go one step forward. It's specific to IgG as indicated here. It will not bind to IgM because there are amino acid sequences that are specific to IgG, specifically a rabbit IgG that you won't find in other species and that you won't find in a rabbit IgM. Yeah.
would it only bind to one specific flavor?
It depends on how that secondary antibody was developed. There are regions of IgG that are common across all those flavors, but then there are regions that are specific to the way they call them is like IgG1, IgG2, IgG3, and IgG4. There are aspects that are unique to each isoform of IgG, but then there are aspects that are common across them as well. And so typically, if it's IgG, if that's all that's listed on your antibody tube or listed, for example, in a question or example I provide, that's going to be a part of IgG that's common across all of them.
I guess I'm just asking, like, it's not assessment, you just need to know that.
Sorry?
Like, if it's on assessment, we just need to know that it's by the secondary antibody binds to this specific one.
Yeah, so for like an example, I want to make sure I don't screw myself here. You could imagine a question that you're provided with a primary antibody and asked which of these secondary antibodies would be suitable. And so the key thing to note would be that whatever this is against, anti-rabbit, matches the species of your primary antibody. You typically would like to avoid you would typically like to avoid having the same species of so if this is a mouse anti-PKAC, you would not want to have, you'd want to avoid mouse anti-mouse because you'd have self-recognition of these antibodies that would be problematic and you'd have false positives of your signal. And so you'd want to have species that are far enough apart where you'd have a separate species that your secondary antibody is originated from that recognizes, that matches, matches the species of your primary. That's another thing to be mindful of. And so when it comes to like what the take-home message is here, it's important to understand the advantages of why secondary antibodies are helpful. And I'm going to go provide a little bit more detail about that on the next slide. But it's important to know how you select if you have a given primary antibody. Sometimes you're restricted by just what's commercially available. Based on what primary antibodies are available, how would you pick the appropriate secondary antibody and why that would be important? Another take home is what the difference is of polyclonal versus monoclonal antibodies. And again, like think back to the example of the hand. If the hand is your antigen, the molecule that your antibody is recognizing, monoclonal antibodies all bind to the same epitope, the same feature, whereas a polyclonal antibody mixture is a mixture of antibodies that bind to different epitopes, different features on that antigen. One example is in the coronavirus, the antigen is spike protein, but the epitope that is of interest is spike protein in its pre-fusion state before it's been before there's binding of spike protein to ACE2, which is the ligand for spike protein. And so there's this very specific region, a very specific amino acid sequence on the N-terminus that our coronavirus vaccines were raised against. And so that specific epitope is what these coronavirus vaccines were raised to recognize. And so the mRNA sequence in these mRNA-based vaccines encode for that very specific epitope on spike protein in SARS-CoV-2. Yes. Conceivably at the same time. They're all binding to just different parts of that same antigen. Yeah. So why are secondary antibodies so important? So we've already talked a little bit about how polyclonal antibodies can be cheaper, but monoclonal antibodies help you detect alterations or expression of a very specific part of a protein. And so that's why polyclonal versus monoclonal is an important consideration. But why can't we just stand with primary antibodies only and just move on from there? So the secondary antibody binds to usually a very specific part of the antibody. Antibodies are typically in a Y shape. the short arm, the upper short arms are where there's antigen binding. Because those short arms are where antigen binding occurs, where we have these like little antigen binding cups at the top, that's called the FAB domain of an antibody. The bottom half is referred to as the FC domain. Again, a helpful way to remember this and distinguish this is that at these antigen binding pockets at the top here, AB, antigen binding, that's called the FAB domain. And the bottom longer arm is called the FC domain. There will be another like helpful trick to understand why that's called the FC domain. The C called it usually stands for crystal, but there's another trick to help remember it that I'll teach you guys in the next class. But the secondary antibodies that we use to recognize our primary antibodies, those are directed at the FC domain. Okay, and so because this is usually a peptide region that's easily producible via like peptide synthesis, these secondary antibodies can be mass produced because antibody sequences are easily have been well characterized. Yes. Yeah, so you're scooping my adaptive immunity lecture. Yeah, so there is in the I'm really fighting the urge to like go like into the nuts and bolts of antibody structure. There is a variable domain and constant domain of FAB. The changes in the variable domain are what allow us to have potentially trillions of antibodies that our body can produce in response to the different numerically possible combinations of amino acids that can be encoded there. And so, yeah, the variable domain is our lives depend on the variable domain being able to change dramatically. We leverage it in research to be able to produce antibodies either for research or for clinical applications in the case of cancer immunotherapy, for example, or immunotherapies in general. But taking a step back and not wanting to like nerd out on that for a moment, the key thing to note here is that the FC domain is what our secondary antibodies are binding to on our primary. So this is the primary, for example. This is what our secondary antibodies on their FAB domain, because their FAB domain is like what they bind to. We'd essentially have, I'll actually use different colors. I don't have different colors. Son of a gun. All right. For a colorblind person, I sometimes like color coding, so forgive me. So I'll just like denote it by a dash. Hit me with that blue. Hope you can catch because I'm going to toss right back at you. So our secondary antibody, and I'll just make like a little key here. Our secondary antibody on its FAB domain is binding to our primary. And it's binding all over because it's largely polyclonal. So there's different epitopes that these secondary antibodies can bind to and are binding all over. And so what's also noteworthy is these secondary antibodies have historically, it's been a chromogenic enzyme, chromogenic meaning like it induces a color change. Oftentimes now we're using fluorophores, but there's usually like some fluorophore that because these can be mass produced and are fairly cheap, we can just use pretty inexpensive reactions to wastefully just try and attach and tether these fluorophores to secondary antibodies. But another key thing to note is that we have amplification of the signal where if we relied on primary antibodies to carry the fluorophore or chromogenic enzyme, we'd have one for each primary antibody. But when we use a secondary antibody, we can amplify the signal. This is one of the key things to note for secondary antibodies. Where on a given primary antibody, we can have, I'm using up your blue, I'm sorry. On a single given primary antibody, we can have many, many secondary antibodies binding that result in amplification of the signal for each primary that binds. And we're going to have lots of primary antibodies that bind to our antigen of interest. And so you just get lots of amplification of your signal. So it can be a key thing to note is that the key takeaways are that we can have this be, and because we can, because secondary antibodies are cheap, because they are easy to just try and couple enzymes and can be it amplifies your signal, you can then try and use less of your primary antibody that's usually more precious and more expensive. So the key takeaways are secondary antibodies can be easily mass produced. They can be easily tethered with these fluorophore chromogenic enzymes. And a key thing is that it results in the amplification of signal, which is really helpful if you're trying to measure some protein that can be hard to detect. I think there were a couple of hands. Yeah.
But when you have like a mixture, and asking for, let's say, right? If that, I think don't get so that.
Wait, so you said you have like a mixture of proteins in a sample that you stain with two species of antibodies. So like one mouse is one and one rabbit.
Treat the primary.
Yeah.
So then if you inject like secondary, since they only target FC general, I mean, right?
No, so it depends on what that secondary antibody is targeting. So in this example, we have two primary antibodies we put in. One is from a mouse and one is from a rabbit. It depends on what that secondary antibody is against. If it's goat anti-mouse, you'd only detect what the mouse antibody is against. If it's goat anti-rabbit, then it would just detect what that rabbit antibody is recognizes.
So like it's general, but still specific.
Yeah. So IgG antibodies in general, the presence of them, the presence of antibodies is true across all of mammalian biology. However, it's not conserved across all species. It's not exactly the same. So a rabbit IgG is going to look different, is going to have a different amino acid sequence than a mouse IgG versus a goat IgG versus a human IgG. And so because is enough difference in the, I'll harken back to something, because there's enough difference in the primary structure, these antibodies can be specific to these species. All right, we can press on. So in the instance that, again, this next slide is just for your edification, not for content that you need to memorize. In the instance that there is and you need an antibody against a certain protein and it's not commercially available, you can contract a company to make an antibody for you. The way in which that happens is there will first need to be, you need to know the primary structure of interest here. And so you'll have peptide synthesis, that's at the top of this quote. And then from there, you will have, you'll have peptide synthesis of that, that's going to be your antigen. Whatever species you want it in this case, it's going to be rabbit. A rabbit will then be injected with that peptide to then induce an immune response against it so that antibodies can be produced. And so in this case, it's going to be polyclonal antibodies. It'd be a lot more expensive if it was monoclonal antibodies because there's the requirement to purify the specific clone of B cell that makes that antibody of interest. And so after 77 days of these mice being immunized with this peptide that's been synthesized, there's then going to be affinity purification. In this case, it's for a phosphorylated version of this protein. And so by and large, for a polyclonal antibody of a protein that where there's not a commercially available antibody, it costs about $5,000, $6,000. If this is monoclonal, it'd be an order of magnitude more expensive. But prices on this are kind of varying with companies being able to refine the process. But this is to help you understand what the process looks like for producing an antibody. Typically, these animals are injected with this peptide, and what animal you inject it in will inform what species your primary antibody is, which will then inform what your secondary antibody is. And so if they had decided that they wanted this for their specific reason to be in a mouse, they could immunize a mouse, purify the antibodies, and it would probably be a little cheaper than using a rabbit because those are more expensive to house. But another key thing to note, at least there's one thing I do want you to walk away with knowing from this slide. While we can purify antibodies, and you can also, there's an effort to want to purify the cells that make that specific antibody, particularly if it's in case of a monoclonal antibody. As you can imagine, B cells, the antibody producing cell, if you collect those, they have a limited lifespan. They're not going to live forever. And so there is a strategy that's been employed to try and make them live forever. Is there any kind of cell that any of you all can think of that does a really good job of growing, dividing, and doesn't stop living, for lack of a better phrase? Yeah.
Cancer.
A cancer cell. So what ends up happening is that a B cell and a cancer cell are, to put it crudely, are fused together to form what we call a hybridoma. So hybridomas are these like B cell, cancer cell combinations where it is producing antibodies. It has that B cell function, and it doesn't die and has that cancer cell longevity. And so we can purify these hybridomas for specific clones to have, in case you're interested in a specific monoclonal antibody. If you only want polyclonal, you can just take a collection of these hybridomas that from the B cells that are activated in response to immunizing the animal with this peptide. And so, again, taking a step back, this is just to help show you how this process works. But a key thing to note is what a hybridoma is, how antibodies are how antibodies can be produced, and that hybridomas are this combination of cancer cells and B cells that leverage longevity of cancer cell and the antibody producing function of a B cell. Yes. It would require a lab to have peptide synthesis abilities. It would require, typically, most labs have mice or rats, and so you can develop a mouse or rat antibody. It would require affinity purification, so some sort of chromatography setup. Those resources are available to one at a university. Even like it's unlikely that one lab will have everything in their space. The resources are available at a given university. It's a lot more convenient to farm it out to someone to do it for you who's really good at it and does it professionally. Because there is the likelihood that if this antibody is so important to you that you don't want to mess it up, and it's easy for, if you're not familiar with doing peptide synthesis, it'd be easy to get that wrong. It'd be easy to get wrong the isolation of sufficient antibodies out of the animal and then to do proper purification. So it's just, a lab could DIY this. 100% yes. It's more convenient to farm it out to a professional. I know that's probably an unsatisfying answer, but for a grad student, it's probably very satisfying for them to hear that as opposed to them having to try and raise antibodies themselves. Yeah.
Like what you do this again?
There's so it would probably come down to convenience, where there was an example where we had a Western blot with ATPase and thyrodoxin where you could measure them simultaneously. If someone's trying to create a large panel of antibodies, you probably already have some that are rabbit or some that are rat and need one that's mouse. And so you'd probably want to go that way. There's nothing inherent to these species. By and large, there's nothing inherent to these species that would make one more advantageous than the other. It is possible that for some protein of interest, there might be significant overlap in the primary sequence of like mouse versus human of some protein. And so we'd say like, let's do mouse, let's raise this antibody in a mouse because we can then use that for human samples as well, because there's enough homology or enough similarity and overlap. So that would be the one exception where there might be an actual biological reason to pursue a certain species. But by and large, it might be informed by, we already have a lot of antibodies that are from these species. Let's try and raise some in these species over here. These are great questions, by the way. All right, we'll go ahead and press on. Again, this is just for you to get a glimpse of how this works. I am not asking you to memorize antibody production protocols, but I will try and highlight what a key takeaway is. So typically, when you're immunizing an animal against some peptide you've had synthesized, what ends up happening is you're going to end up basically creating almost like a vaccine with this peptide to elicit an immune response and elicit antibody production. So on day 0, you'll have your rabbit, in case you're doing this with a rabbit, you'll pre-bleed so that you can see what the antibody levels or antibody titers look like before you immunize them. You'll do a subcutaneous injection of your first exposure, and it is what's called with Freud's complete adjuvant. What that looks like, excuse me, is whenever you hear adjuvant, think of a molecule or collection of molecules that help elicit an immune response. So Freud's complete adjuvant, or FCA, what that is a mixture of mineral oil, surfactant, and dead mycobacteria. The mineral oil helps to maintain a certain viscosity so that when this is injected subcutaneously, that's what the SC is here, that it doesn't immediately diffuse away. The surfactant changes it contributes to maintaining the local exposure of this antigen within that local site. And the dead mycobacteria is basically a way to awaken your immune system and trigger a really strong immune response while this peptide is available. And so whenever you see Freud's complete adjuvant, which has dead mycobacteria, think of that as a sledgehammer on your immune system to wake it up immediately and to trigger an immune response to this antigen that's being exposed. And so historical vaccines typically was a combination of when you would have a first booster, it would be with complete adjuvant in some way. And then secondary boosters would be incomplete adjuvant. And so Freud's incomplete adjuvant has no dead mycobacteria or no strong immune eliciting reagent. I don't want you to think that the current vaccines that we get have a mixture of dead mycobacteria, but rather we'll have some sort of factor that triggers a strong immune response, which is sometimes why people will feel a little under the weather when they get a vaccine, because you're triggering an immune response that elicits your immune system to wake up. So while that feels pretty crappy, what ends up happening is that you've awakened your immune system to then elicit a response to the peptide or the antigen that's being delivered. About 3 weeks later, day 21, there's another booster with incomplete adjuvant, no mycobacteria. Another 3 weeks later, day 42, another booster. And then about 10 days later, there's going to be a bleed where there's going to be blood collection and able to see what the titers are, again, what the levels are of antibodies that have been raised against your antigen that you've introduced here, in this case, the peptide. Yes. Well, sorry, great question. I should have clarified this. Freud's complete adjuvant and Freud's incomplete adjuvant both have your peptide of interest here in them. I appreciate you asking that. I should have made it more clear. In the case of antibody production, both the complete adjuvant and the incomplete adjuvant will have your peptide of interest, because that's what you're trying to elicit an immune response against. Some of the historical vaccines, there's going to be whatever the antigen is, whatever that virus is also going to be present in the complete adjuvant or incomplete adjuvant. And so again, in this case, you're going to have the Freud's complete is going to be your peptide, mineral oil, surfactant, and mycobacteria. Freud's incomplete adjuvant is going to be your peptide, mineral oil, and surfactant only. And so you'll collect a blood sample to look at titers. You may need to boost again with another round of Freud's incomplete adjuvant, take titers again, and then euthanize the animal. And so what we typically do for being able to measure the antibodies to make sure that they can bind to that they're actually binding to our peptide of interest is we do something called a direct enzyme-linked immunosorbent assay. Again, immuno means antibodies are involved. Another way of calling this is called a direct ELISA. So some of you may have heard of ELISAs, and so this is one form of ELISA. This is a direct ELISA. So what ends up happening is that you coat the bottom of your plate, your well plate, with that antigen. In this case, it's going to be some peptide that you've synthesized that you're going to coat the bottom of the plate with. And when we talk about coating the plate, there are what we call high binding plates that are treated and enabled that are more likely to bind via electrostatic interactions to your peptide that's been synthesized. You'll block because we don't want non-specific binding. You'll add primary antibody. In the case of antibody production, you might take different hybridomas and the antibodies they produce and use them in different wells to see which one binds best to our peptide of interest. And so that's in the case of antibody production. In the case of direct ELISAs in general, we'll just refer to it as your primary antibody. But just to help you understand, like, this is how they test which antibodies that have been produced best bind to the peptide we have here. So you have your primary antibody that you incubate with. There's always washing steps in between all of these. You'll add your secondary antibody that is, again, raised against, that recognizes the species of your primary antibody. And the secondary, usually in the case of an ELISA, will be enzyme linked. It will have some sort of chromogenic enzyme that reacts with a substrate to produce a color change. You add that substrate to the well. It's usually a solution that you fill the well with. And if that secondary antibody is there, that enzyme is there, it'll use that solution as its substrate and induce a color change that will be directly correlated to how much antibody binds to your peptide or your antigen. And so you just measure the absorbance or the optical density of the well. Another form of an ELISA is called a sandwich ELISA. This is one that is routinely used in labs on a regular basis. Was just in my lab troubleshooting one with a student just before I got here. And so the way this works, and you'll see why it's called a sandwich ELISA in a moment. Instead of coating the bottom of the well with a peptide or with your antigen, you coat the bottom of the well with an antibody. In this case, we refer to it as your capture antibody. And so again, these are treated plates that are able to bind to proteins. You block to reduce nonspecific binding. And then usually you have some sort of sample that's a mixture of proteins. With a sandwich ELISA, you're trying to measure and detect one specific protein. So this could be any kind of sample, whether it's blood from a patient or from a mouse in an experiment. It could be the media that your cells are in and trying to see if they secrete a certain protein of interest. And so it's some sort of solution that's a mixture of proteins. You'll add that to your well, you'll incubate, you'll wash again, and then you'll have some degree of binding of that protein that's in that sample to your capture antibody. The reason why it's called a sandwich ELISA is because you'll then add another antibody on top called detection. And so it's basically a sandwich of antibodies in the well. Excuse me. There's going to be, I should have changed this. Instead of having another third antibody, what will usually happen is that this detection antibody will have some sort of molecule on the Fc domain of it. Oftentimes it's going to be biotin. And the reason why is because we leverage the fact that biotin is really easy to, this isn't critical for you guys to know, but I enjoy talking about it, so I'll share the information. Biotin is really easy to tether or add on to proteins. And what it binds to is avidin, and that is the strongest affinity known in nature between those two molecules. And so we leverage that really strong affinity to do either sandwich ELISAs or lots of other scientific research approaches. But usually that detection antibody will be have avidin or biotin on it, and then you'll incubate with, instead of enzyme linked secondary antibody, we'll say this is avidin with an enzyme on it. And usually that's an enzyme that reacts with our substrate solution. You'll then add your substrate solution. You'll get color change based on how much signal is produced, which is again based on how much sample binds to your capture antibody. And you'll measure the optical absorbance or optical density. So these different, sorry, go ahead.
Why would you do this if it provides? We'd like to use more antigens here.
Yeah, so it all depends on what, again, I'm gonna keep coming back to this phrase, but it depends on what question you're asking. So if you're trying to measure the presence of a specific protein in some sample that's a mixture of proteins, sandwich ELISA is the way to go. If you have if you are trying to answer how well, let's see the way to describe this. If you're trying to measure for perhaps are there antibodies that are produced in response to some sort of antigen or some molecule, and you're trying to understand, like, is there an immune response to this? Is the body making antibodies against this molecule? One example that's trying to be answered currently in the field of biomaterials is, for example, PEG-based or polyethylene glycol-based biomaterials. Is our body making antibodies against PEG? Like, we use PEG for so many different things, but for some reason biomaterials keep inducing a negative immune reaction in patients. Are we inducing a response against PEG? And so you would coat the bottom of your well with PEG and try and see, are there antibodies being produced against it? And that would be a time to use a direct ELISA instead. And so if you're trying to understand, are there antibodies being produced in response to something, you want to go direct ELISA. If you want to know how much of a sample is present in this mixture of proteins, whether that's in blood, cell media culture, some sort of patient sample like sputum or bronchoalveolar fluid, you'd want to go sandwich ELISA because that'll tell you how much is present in that specific sample.
So it's concentration versus like if it's there.
Concentration versus is there interactions between I guess it would be twofold. For this, it could be the scientific question could be related to the coded antigen or in relation to this primary antibody. You could be answering, is this antigen actually inducing an immune response? Is it antigenic? Like, is it actually inducing an immune response? Or are these antibodies sufficient to bind to this specific antigen? And so it's more antibody focused usually with direct ELISA, whereas with a sandwich ELISA, the question is more related to expression of some specific protein of interest. Great question. That actually leads quite nicely into my next slide, where ELISAs are really, whenever it comes to thinking about how to approach an experiment, there's always some considerations to consider. There's always time, there's always cost, and there's always how quantifiable something is. As engineers, we always want something to be quantifiable. We want to be able to measure things and have a way of doing statistical analysis on it to determine if there's a significant difference or not. So with ELISAs, it's much more quantifiable than, say, a Western blot. where you're looking at a band, and usually it's a little more qualitative. There are ways to make it quantifiable, but it's not as high throughput or quantifiable as ELISAs. So the question is, why wouldn't we just do ELISAs all the time? Which for some people would say that's what we do all the time. But there's a reason why sometimes ELISAs might not be the best case. And again, it all comes back to what question you're perhaps trying to answer. And so one example here is that you can use ELISAs to measure proteins that are inside the cell. For example, this protein called ERK, ERK12, there's different isoforms of it, which I'm pointing at when the screen goes out. You can, in this case, there's exposure to Coxsackie virus where they're trying to measure ERK activation. And so when you're doing an ELISA, it's really quantifiable. You can get really great gradient of expression and detection. However, the antibody used to measure ERK, also to the top right here, it's usually meant to measure two forms of ERK or two isoforms, ERK1 and 2. That same antibody actually binds to ERK5, which would have been totally missed in the ELISA because that otherwise wouldn't have been known, because there's this assumption that this only binds to ERK. When this antibody was first produced by a commercial vendor, that region of ERK1/2 is also conserved in ERK5. And so that's something that would have been totally missed with only using an ELISA, where there's this presumption of only being able to see ERK, because usually ERK has a molecular weight of 42 and 44. That's why there's two bands here. And ERK5 has a much higher molecular weight. So being able to separate them out by their molecular weight and see them differently will enable this group to see that distinction. And so they're able to see that in response to Coxsackie virus exposure, that when using just an ERK1/2 inhibitor, it doesn't really affect the activated ERK signal. But when you use an ERK1/2/5 inhibitor, it totally dampened it back to near background levels of ERK activation. So back to, in response to the question that was asked on the last slide, there's always the question of why would I use this technique versus this technique? It all comes back to the question you're trying to answer. It also comes back to what information that approach or technique gives you. And so, again, the ability to separate based on molecular weight at times can be really advantageous when it comes to Western blotting, despite it being something that usually takes about 3 days long to do, whereas an ELISA usually takes about 4 or 5 hours to do. But again, there's kind of a cost-benefit analysis there. I'll stop for a couple of questions there. Yes.
Wash before you add the chromogenic substrate, because otherwise wouldn't the absorbance like measure all of the antibodies there, not just the ones that did?
Yeah, so this is a little reductive. So if you want to know what an actual ELISA protocol looks like, again, I didn't want to try and overwhelm. After you block, you typically wash. After you add antigen, definitely wash. After you add your detection antibody, wash. After your enzyme-linked secondary, wash. And then you add your chromogenic substrate. So by and large, almost between every step, you need to wash, because you want to reduce the possibility of there being accidental binding of molecules where you otherwise wouldn't want it to happen. I just didn't want to inundate you all with wash steps. I'd also say that for those of you like getting into labs, it's always great to like learn one specific skill right off the bat that like makes you valuable and helpful. This was the first skill I learned. I would just crank out ELISAs all day long to a point where it became a game of like, how many ELISAs can we do simultaneously in a given day? Got up to 12. That became a miserable experience. I don't recommend it for any of you. But to try and relate this back to like research and perhaps some of the experiences you guys are doing, ELISAs are a great first technique to learn. They do take like 4 or 5 hours, just because like each of these steps is almost like, the block, the antigen, the antibody steps are each 1 hour incubations. So like you can do a blocking step, prep your next step, do your detection, go on a run, come back and do your your next antibody step. But the key thing to know is that this is super accessible and super doable. It looks a little intimidating when you see it like this, but is a great first skill to learn for any undergrad trying to get into research or trying to get their foot in the door. I'll leave space for a couple more questions there. And again, if you're worried it's a dumb question, I love dumb questions because they're really easy to answer. And I also wouldn't call them dumb questions because sometimes they can be really fundamental questions that actually get to the heart of something that I didn't touch on. Yes. It's kind of great question. It's kind of both and. So the to what's written on the board here, primary antibody is rat antitubulin. So for this antibody to be created, it was a tubulin peptide that was injected in a rabbit, and this was the antibody that was raised or antibody that was developed. So it's the species of that antibody is rabbit. So for your secondary antibody that you want to use for it, you wouldn't want to use also a rabbit. That you'd lead to problems and cross-contamination of your antibodies. You'd want to use some other species. Typically, it's goat. That's always a safe bet. It's widely available. Goat anti the species of your primary. So this antibody was injected in a rabbit. Its species is rabbit. And so you'd want anti-rabbit antibody or anti-rabbit IgG. Yes. And if I didn't make that explicitly clear, that I'm glad you said that. Goat anti-rabbit will bind to any rabbit antibody, regardless of what that primary rabbit antibody recognizes. Because again, it's just binding to the Fc domain, this long arm of that rabbit antibody. So you can use and typically buy them in large quantities, where they will typically bind to any rabbit antibody, any antibody raised in a rabbit, and that the antibody is identified as a rabbit anti whatever it is. Thank you for saying that. I appreciate you asking about that. Yes. Yeah, so there's two main differences. One is the order of operations of how you use them. And number two, typically in most ELISA kits that you buy today, that detection antibody is conjugated or tethered or tagged with biotin. And so that's another one of the main differences between detection versus capture. Sorry? When you use them during the process. Yeah. Yep.
I think counting like that.
Mhm.
You said that. Secondary also.
Yeah, so if you did a Western blot and you were detecting for both, you wanted to detect for both PKA and PKC. If you used rabbit anti-PKA and rabbit anti-PKC, and you used goat anti-rabbit as your secondary, you could not distinguish the expression of those two proteins on your membrane. It would look like just one big old band. And so what you'd want to do is use different species so that you can then stain them at the same time. You can have a mouse anti-PKA and a rabbit anti-PKC. Because they're different species, what you can do is you can incubate those two antibodies together, but then do your secondaries depending on what's attached to your secondary antibodies. If it's like different fluorophores or different wavelengths, you can throw them both in at the same time too, because they have different wavelengths, and so you can detect them separately. And so if you're using the same species to measure two proteins, the only way that can work is if you're doing a Western blot. I'm going to go back quite a bit here. Forgive me, almost there. The only way that can work is if their molecular weight is far enough apart. So for example, if thyrodoxine is around, let's say 20 kilodaltons, ATPase is right around 50, that's enough separation where you could use the same species. That's fine. Because a Western blot separates by mass or molecular weight. Totally fine here. If it was even higher at 100, like that'd be great. A rule of thumb for me was always, it needs to be about 40 kilodaltons apart, because you'll sometimes have smearing, depending on how good you are at Western blotting. You don't want to try and measure any proteins that are closer than 40 kilodaltons apart. If you're running an ELISA, you couldn't, there's a new approach to ELISAs that tries to detect for multiple things at once, and it's a bunch of different antibodies that bind to different things. That's called multiplexing. That's a little more complicated that I'm not going to get into, but it leverages different species of antibodies that bind to different things. Yeah. All right, so we are in the home stretch. I'm just going to go ahead and advance back to where we were. So like Western blotting, like a sandwich ELISA, and like affinity chromatography, we're going to get back to the anthrax and pregnancy test subject that was our application point. Lateral flow immunochromatography. Immuno, there's an antibody involved. Again, whenever you see immuno, think antibody. So with lateral flow immunochromatography, whether it's a pregnancy test, an anthrax test, or the SARS-CoV-2 test, what ends up happening is that you have usually a sample port where you load your sample. And then we have antibodies attached with these gold particles, excuse me, that then, again, lateral flow, they flow across a resin. And we have these capture antibodies that will also go through. And so if you can recall for some of these tests, that there's both a test band and a control band. Control band is to make sure that the test is working properly. And what that looks like is kind of similar to a direct ELISA. You kind of have a direct ELISA and a sandwich ELISA happening at the same time here. What measures your test sample is the binding of whatever was in your, whatever biosample you introduced that binds to this capture antibody. The control strip only works when the antibody that's tethered with the gold particle binds to the given antigen that's stuck at that control strip test site. And so to make sure that the test works, to make sure that antibodies are flowing across this resin, you should have antibodies binding, gold particle tethered antibodies binding to this control strip site. And that will introduce the control band that you typically see in these tests. And this sandwich ELISA will help determine whether or not you test positive for whatever that given test is. And so the bands that you typically see is this concentration of either a gold particle or some other particle that's visible, that is within the visible spectrum, is the accumulation of antibody, accumulation of signal that then creates this positive, this band that you see that is the positive strip. That'll either be like a single band or for some tests, a positive band that shows up because there's antibodies that are in that shape that then have the gold particle that shows up with your capture antibody. And again, if this is what it would look like if the control strip, if the test worked, you'd have a positive band at the control strip and a no band at the test. If you like biochemical methods, we have a cohort of folks here at UVA that are actively trying to push the limits of some of these methods, especially within our department. Both Kevin Janes and Tom Barker developing them both for either diagnostics and therapeutics or for new biochemical methods, as well as others in chemistry. And so with that, I'm going to go ahead and call it. Thanks for your time. And I hope, emphasis hope, I'll be able to see you guys on Thursday.